Good morning, everyone, and welcome back to the virtual international conference on global research trends in biotechnology hosted by the Department of Biotechnology from St. Joseph's College of Engineering. It gives me great pleasure to introduce our esteemed guest speaker for today's session, Dr. Nirmal K. Sampathkumar. He graduated with the highest distinction in biotechnology from Anna University in the year of 2011. He then did his master's in biomedical engineering at Université Paris Descartes and went on to pursue his doctorate in neuroscience from the same university. He was a master research intern at the same university as well as at Université Paris Diderot. He went on to be a research intern at other prestigious institutions like University of Zurich and INSERM. He then became a postdoctoral research associate at USC Leonard Davis School of Gerontology. He is the recipient of many awards and scholarships, including the Paris Tech Foundation Scholarship. He is a patent holder and has published papers in various journals, including in the PNAS. Dr. Nirmal has also been an active member of Raise Rural and aids in spreading research awareness amongst rural students. In this community, he was awarded the Young Researcher Award in the year of 2010. He is currently a postdoctoral scientist at King's College London, which is one of the top 10 universities in the world. Sir, it is an immense pleasure to have you with us today, sir. I request you to take over the session.
Thank you very much, Swadika, for this wonderful introduction. I feel really privileged to take part in this wonderful conference that is really conducted in this crisis time, which is almost impossible. So thanks to you all for conducting this wonderful conference in the first place. And I also I extend my warm thanks to my mentor back in Aryl Engineering College, Professor Parali Subramani, through who I got the contact of Professor Chamundishwari. So thank you both for giving this wonderful opportunity. And to be honest, I'm really young, and I don't know if I have enough thing to take part in as a speaker in this fantastic event. Thanks for that. And without further ado, I'm gonna start sharing my screen and then share a few of my works. So for this particular session, I'm gonna walk with you guys some of my key findings during the past few years during my PhD, as well as my first postdoc in the Benoit laboratory back in USC. So this particular presentation will have like 3 different sections. This is the summary of my talk, where I'm going to discuss Aryl hydrocarbon receptor, its discovery, and a brief introduction what is AHR is. From now onwards, I'm going to call Aryl hydrocarbon receptor as AHR. And second section of this presentation will be, I'm going to cover AHR's role in peripheral nervous system, particularly in terms of like myelin development, myelination, and also its potential role in peripheral nerve tumor. And the last section of this particular talk will be AHR role in macrophage aging, and I'm going to explain like what is macrophage aging and its AHR's potential role in sex dimorphism in aging. So to briefly give the discovery of AHR, it's a very interesting story. We have to go back in 1976, where it was first described as an induction receptor by Polam et al. So what they saw is that after adding dioxin, they saw some of the So they saw some of the enzymes like aryl hydrocarbon hydroxylase and other enzymes, induction in the enzyme activity after adding TCDD. And then they thought that there could be some receptor involved in this induction of these enzymes activity. So they called it as induction receptor back then. Since then, after the discovery of AHR, it is called as dioxin receptor. And quickly introduce about the mechanism, the AHR pathway. So AHR is a ligand activated transcription factor commonly found in the cytosol. And binding with the ligand, it translocates and sheds its chaperones proteins and then heterodimerize with ARNT in order to initiate the transcription of its target genes, CYP1A1, for example. So one such ligand would be TCDD, one of the potent ligand, an agonist for AHR, which is a very toxic element. And AHR is acts as a receptor, and end of the process, we have the CYP1A1 and other cytochrome P450 enzymes involved in metabolizing these xenobiotics. If you look at the domains present in the AHR protein, we can see that it has the BHLH, making AHR belongs to the BHLH superfamily. It has two PAS domains, and it has ligand binding domain and transcriptional activating binding domain. So when we look at the there's like several endogenous receptors that are described in the literature, and as we can see here, tryptophan metabolites is found known to be like endogenous ligand for AHR, and L-carnitine draws the main attention because we have this endogenous enzyme that can convert tryptophan to L-carnitine, and there's like bunch of paper. The first paper that described Aryl hydrocarbon receptor endogenous ligand L-carnitine could be a potential cause for some tumors, and this particular paper also discusses that it could be involved in inducing the hold on, sorry, I want to move this somewhere else because it just hides the title. Anyway, sorry. So it's just involved in tumor progression. So moving forward, and when we talk about the AHR conservation in the evolution, we can see that the AHR homologue found to be discovered at the level of chordata all the way to arthropoda in invertebrate systems. And in terms of the invertebrate system, what is this endogenous role? So when we look at the evolution, so AHR is found to have this homologue called spineless in Drosophila that is first described in 1975, and it has a huge role in the retinal mosaic formation and also dendritic arborization and also neuronal development. So when we have the spineless mutated, we have the dendritic arborization disturbed and also the neuronal development also disturbed in Drosophila. So when we look at these protein domains again, please make a note that the ligand binding domain is also exists in the invertebrate system, but for some reasons, all the ligands that are described in the literature so far that binds to the vertebrate AHR, not so far shown to have any interaction with the invertebrate homologue of AHR, meaning that this particular protein acquired this kind of new xenobiotic metabolism function with the evolution, whereas this particular protein has other endogenous roles. As you can see here, when you knock out AHR homologue, we have problem with the neuronal development. So this until like let's say early 2000, everybody was working with toxicology aspect of AHR, but these works draw attention to a lot of researchers in terms of like what's the other role of AHR. So basically AHR is not just a dioxin receptor. When you look at the literature, the accumulating evidence show that AHR is involved in central nervous system development, heart development, liver, kidney, different things. And when one of the teams of my host lab also showed that when you knock out AHR, we have problem with the oculomotor system. And another team from our lab also showed that AHR ablation could lead to demyelinating kind of phenotype and also inflammation. When we look at the tumor, AHR behaves differently in a cell-specific manner that in some cases it involved in the tumor progression, and in some cases activation of AHR could be involved in tumor suppression, these kind of things. and we don't know. It's a cell-dependent manner. So if you look at this overall picture, AHR is known to be involved in several functions. I mean, xenobiotic is one of the main things that well documented, and something is missing. And in that case, that's where I came in. So we asked the question that what could be the role of AHR in peripheral myelination and peripheral nerve sheath tumor. So we have a reason to ask this question, and I'll explain in a while. So for this particular work, I was doing a majority of my research in this beautiful, beautiful, beautiful building from outside, of course, in Charbel's with Charbel's team back in Paris. So the second section of this talk, we're gonna see what's the role of AHR in peripheral nerve system, especially in myelination. So to quickly introduce you what is the importance of myelin, several of you might know already what is a myelin is, so let's not go deep into it, but myelin is important for the nerve conduction and for the salutary signal conduction in axon. And this particular myelination is basically an extension of Schwann cell membrane in terms of like peripheral nerve system, whereas this is achieved in central nervous system by oligodendrocytes. So if we take a deeper look at the myelin lamellae, so we have this different lamellae that are compacting on each other to form this nice myelin sheath. And in this compaction, the major membrane proteins involved are namely P0 and PMP22. So P0, myelin protein 0, PMP22, peripheral myelin protein 22. And any problem with this imbalance in the P0 and PMP22 protein proportion could lead to some kind of compaction problem and eventually lead to nerve conduction problems. And quickly to show you how these neural crust cells are differentiated to myelinating Schwann cells, so there are several pathways involved pushing or progressing towards this positive or not positive, like matured state. So for example, neuroglin 1 is involved in like when you have this positive activation of neuroglin 1 pathway, so Schwann cell precursors are turned into immature Schwann cells, and eventually there are other pathways, activation of those pathways could positively drive towards the maturation of Schwann cells in making myelin. And we have demonstrated that AHR is involved in myelination, as well as it could potentially be involved in malignant nerve sheath tumor, and perhaps blocking AHR pathway could be beneficial. So in this section, we're going to quickly discuss the key results. And for example, here, knocking out AHR in mice led to some motor deficits. As we can see here, the knockout AHR did not show any improvement over the days, whereas the wild type improved the learning, so motor coordination. So as you can see here, AHR led to some motor deficit. So this made us question like what could be the reason, and then we designed an experiment based on the is it due to some myelin problems? So we used animals like complete KO and the Schwann cell specific KO, and I'm not going to discuss the Schwann cell specific KO today due to some time constraints. So and also in the developmental aspect, I'm not going to discuss like the developmental side of the myelination as well, but quickly we're going to see what happened with the myelin in the animals. In that case, what could be the mechanism? In terms of like the mechanism, we harnessed the MSC80 cell line, which is a Schwann cells cell line, and I have silenced AHR in order to demonstrate the mechanism behind this myelination process. And finally, what could be the potential role of AHR in tumor that is particularly related to Schwann cells. So plexiform neurofibroma and MPNST are two different neurofibromas. So one is benign and the second one is the malignant form. So first, in order to do that, we have to check if at all AHR is expressed in the peripheral nervous system. As you can see here, the sciatic nerve, we have the expression of AHR, and we also checked in the MSCAT, the cell line that we use. So AHR is clearly expressed in these models that we used. And then when we move forward, so this is a sciatic nerve. So basically the nerve that runs from your spine all the way like through the thigh region. We extracted, I extract the sciatic nerve and did some biochemistry and show you that P0, PMP22, the major membrane proteins involved in myelin compaction are significantly increased with AHR ablation. And when we checked what's the thickness of the myelin with electron microscopy, we need to calculate the G ratio. Basically, you get the axonal perimeter and divide it over myelin perimeter, and you get this number. And as you can see here, increased G ratio significantly, meaning that the myelin thickness is reduced with AHR ablation. We also checked the axonal perimeter. There is also significant increase in the axons with AHR knockout. So we wanted to know what could be the mechanism. So what I did is silenced AHR in MSCAT. So silencing AHR led to increased P0 protein level, intriguingly reduced PMP22, and we also checked in order to see what's going on with the transcript level. I checked P0 and PMP22 mRNA levels after silencing. As you can see here, P0 and PMP22 levels are increased significantly. And I also wanted to check if it is happening even at the promoter level. For this purpose, I've used P0 luciferase and PMP22 luciferase construct. And as you can see, silencing AHR increased the transactivation of P0 as well as PMP22. So all this put together from the MSCAT side, when we don't have AHR, looks like there is an activation of P0 and PMP22 genes even at the promoter level. So one could think that AHR, given that AHR is a transcription factor, it could be involved in increase these genes directly by recruited at the promoter of these genes. So we used softwares. JJ did this Mat Inspector thing and then found one potential binding site for PMP20 and 4 different potential binding sites for P0 promoters using Mat Inspector. And what I did is that I did a chip qPCR where I pulled down AHR, CYP1A1 being the positive control and histone 3 as a positive control for if the chip worked, and IgG as a negative control, as you know. So as you can see here, it worked, whereas we did not see any recruitment of P0 or PMP22 on any of those potential binding sites. So I was wondering what could be if AHR is not recruited on the myelin protein, what could be the mechanism? And I went back to the literature. My own lab already showed, Charbel and this team already showed that Wnt beta-catenin pathway is a direct driver of myelination, peripheral myelination. So left TCF, so basically recruited at the level of P0. MPZ is P0 as well, you know that. So PMP22 level, and then I was like, is there any crosstalk between AHR and beta-catenin pathway? And there's bunch of articles showing that AHR and Wnt beta-catenin pathway could have some crosstalk. And this PNAS paper is a particular drew the particular interest because this particular paper showed that AHR can have some, there is a PPI, protein-protein interaction. We don't know if it is direct interaction, but there could be a protein-protein interaction between AHR and beta-catenin. Moving forward, what I did is basically I did a co-IP. I pulled down beta-catenin And as you can see here, and then plotted for AHR, and clearly there's a AHR and beta-catenin interaction at the protein level. Further, I also wanted to see, okay, after silencing AHR, is there a change in the level of beta-catenin? So total beta-catenin is also elevated significantly, and active beta-catenin as well after silencing AHR, meaning that AHR silencing could lead to active Wnt beta-catenin pathway activation. In order to check that, I took some of the downstream, not the downstream, basically, some of the components of Wnt beta-catenin pathway, and check the transcript level of LRP signal, LRP, like the starting from the core receptor and all the way up to like target gene, which is axin 2. Pretty much all of these components of Wnt beta-catenin pathway is elevated significantly with silencing AHR, meaning that when you silence AHR, Wnt beta-catenin pathway is potentially activated, given this hallmark axin 2 is also elevated. And then I wanted to check if even if the transcript, at the promoter level, so basically super top flush, STF luciferase is basically a construct that has several binding sites for TCF-left, and silencing AHR also significantly increased the transactivation of super top flush, meaning that a Wnt beta-catenin pathway is activated. So, and further, one could now think that, okay, so now, since we know Wnt beta-catenin pathway is involved in myelin genes, transcription and silencing AHR could lead to Wnt beta-catenin pathway activation. So what I did is I did a chip qPCR again. So I silenced AHR. I pulled down beta-catenin and checked the recruitment of beta-catenin on the level of P0 and PMP22 promoters. As you can see here, that two binding sites of P0 have significantly increased the recruitment of beta-catenin, as well as PMP22 after silencing AHR. So we know that AHR silencing induced beta-catenin recruitment on myelin genes promoter. That part of the story is concluded that AHR is crucial in myelination. And moving forward, to give a brief introduction about the neurofibromatosis, NF1. So NF1 is a tumor suppressor gene. As you can see, it is involved in regulating major pathways that are involved in cell proliferation and cell survival. As you can see here, so MEG pathway, RAC pathway, mTOR, PI3 kinase, AKT pathways. And neurofibromin is necessary for normal functioning of the cell when cell proliferation, cell survival. And NF1 is an autosomal dominant human disorder that has some mutation in NF1, and this is the incident rate is 1 in 3,000. So which is, let's say, it's not that common, but still 1 in 3,000 is not that rare. So people having this particular mutation, this particular gene could have some kind of clinical manifestation. 20 to 50% of the people may develop a plexiform neurofibroma, which is a benign tumor. As you can see, it's just like it's not fatal, but it gives some I don't know how to say, like aesthetic. There could be some aesthetical issues with the tumor. Whereas MPNST is a benign form. So this NF1 primarily composed of Schwann cells, and the treatment options could be like surgical removal of those tumors with and without of different combinations, for example, radiation therapy or chemotherapy. And MPNST is the malignant form of this one. It has very poor prognosis and there's no effective treatment. And this benign form of tumor to the malignant form needs some further genetic lesions. For example, if you have a mutation only in NF1, So malignant transformation needs further genetic lesion. So if you have mutation in NF1, doesn't not necessarily mean that one could develop the malignant form of this nurtured tumor. So there are several literature showing that there needs to be another gene involved or several other genes involved in this malignant transformation. So in order to see that, we collaborated with Eric Passmo in the Curie Institute, and we first of all checked like what are the composition of this tumor. And as you can see here, it has like Schwann cells, mast cells, and endothelial cells. And as I've already mentioned, like the primarily composed of Schwann cells. And we checked the expression of AHR transcript, like the AHR pathway transcript. As you can see here, it is highly expressed in Schwann cells. And the tryptophan metabolizing enzymes also expressed, okay? And then we asked the question, what could be this expression level in the tumors? So being the dermal neurofibroma, basically the neighboring tissue being the control, we checked AHR, the endogenous repressor of AHR, different enzymes that are involved in metabolizing, involved in making the endogenous ligand for AHR. So S100BP0 is being the marker of Schwann cells, and K67 is marker of proliferation. So you can see here that plexiform neurofibroma, which is a benign form, you can see already that AHR shown to be significantly increased, and the repressor of AHR is significantly reduced, and the endogenous ligand-making enzymes are also upregulated significantly. And further, when you look at the malignant form, it further increased, the AHR is further increased, and you can clearly see that the huge increase is the endogenous ligand making enzymes, meaning that there could be possibly AHR pathway is activated in these tumors. So what I did, I took some of the MPNST cell lines, and I just showed you, for simplicity reason, just one of the things is called STS26T. What I did, I inhibited the AHR using just, I show you just one antagonist, a TMF, with 10 micromolar. As you can see here, inhibiting AHR could lead to increased annexin 5 staining and PF5 staining, meaning that the cells are entered into apoptosis, and also finally they're like dying. So, and as you can also see here that this is a positive control that TMF is working, that the target gene CYP1A1 is significantly decreased. And in order to show that even further that it's not something random, we wanted to use specifically silence AHR with SAHR, and as you can see here, the AHR, silencing AHR resulted in increased apoptosis based on, in a dose-dependent manner. So 10 nanomolar and 20 nanomolar increased the PI staining. As you can see clearly that this quadrant have more cells. So to summarize this first section of, the second section of my talk is that, so there's a balance between AHR and beta-catenin activity, let's say AHR and beta-catenin pathway activity in the normal physiology. And the organism is happy, or at least a particular tissue, there's no problem. In terms of imagine that we ablated AHR, so there's an elevated levels of beta-catenin pathway that could potentially lead to myelin genes dysregulation. So there's a problem with the myelin genes transcription that could lead to thinner myelin and locomotor defects. In terms of the pathology, for example, so AHR plays a crucial role in, let's say, the balance between cell survival and cell proliferation. Imagine there's this NF1 mutation that could already lead to a pathological form where there's a loss of balance in this thing, and we have this tumor formation. And in addition to this NF1 mutation, imagine there's an increased expression of AHR that could potentially contributing in the transformation of malignancy, like malignant transformation of benign tumor to a malignant one. So that's the conclusion of part one. And the section 3, we're going to see AHR's role in macrophage aging. For this, this piece of work is carried out in this beautiful building, of course, from outside. So in Bernice, with the Bernice team at the University of Southern California. So as we can see here, I was talking about cancer, and cancer is also one of the things that also age related. As you can see here, it tends to increase with age, as we all know that, including other diseases like heart disease or diabetes or any neurodegenerative diseases. And we all know that we all age, and aging is inevitable. And there are a lot of problems, as I mentioned earlier, could be related to aging. And thanks to Professor Essa, who's a keynote of the who delivered his speech on first day, that he also talked a little bit about aging. So I don't. He already set a stage for me there. So in terms of like aging, there's like at this point, there's like 9 major hallmarks of aging that is described in the literature. You can see here different of them. And Bernice, the Benin laboratory, was primarily focusing on genomic instability, epigenetic alteration. And though they also work on other aspects of aging as well. And when we talk about the aging, there's a new trend started early this decade, that this inflammaging. So basically, age-related inflammation, a sterile inflammation that's going on with the system as we age. And when you look at this inflammaging, so immune system plays a crucial role in this whole inflammaging. And as you can see here, that immune system could be categorized into 2 things: innate and adaptive immunity. And there are several cell types that are involved in doing this. There's 2 different wings of immune system, and we're not going to talk about adaptive immunity. So we focused on the innate immunity as it's the first line of defense, and particularly about the macrophage. Why macrophage? Because it's one of the first few cell types that are involved in responding to any kind of injury, and also clearing the cell debris in terms of like after an injury or with also aging. So when you talk about the macrophage, so the diversity of macrophages is huge. Basically, we have the macrophage that are bone derived. So basically, we have the bone marrow, and we have the hematopoiesis going on. We have the monocytes, and the monocytes could be differentiated to the macrophage or dendritic cells. So we have that throughout our life. At the same time, there are tissue-resident macrophages. For example, microglia in CNS derived from the yolk sac, and we have other tissue-resident macrophage based on the tissue type. They have different names, as you can see in this other F. So, and macrophage are one of the major phagocytes involved in clearing up any debris after an injury or any other insult. Sex as a biological variable. Why sex as a biological variable is that it's a sad truth that a majority of the work, majority of the literature, being preclinical, even at the clinical level, do not include the other sex, specifically, sorry to say, females, in the study, and that's a huge problem. I mean, we are we're talking about SpaceX, Elon Musk, and then we are like talking about tailored or like individualized, specialized medicine for each person. And even today, other like the other sexes not particularly ignored, but due to some reasons, it is not involved in studies, which is basically the other F population of the world is not involved in included in the studies. So in majority of the studies, so which is a sad truth. And in that case, it's also recent days. So sex is also involved. So basically, we want to know what's going on fundamentally. What's the difference between males and females in terms of like inflammaging or these kind of things, in order to understand the basic difference in terms of like cellular physiology of different cells. So this particular cell talks about the estrogen receptor regulate the innate immune cells and other signaling pathways. And this particular cell is a very interesting one that, for example, microglia from females act differently after insult compared to microglia from male mice brain. So in that case, in Bernie's with Bernie's team, I mean, in the Benign lab, what I did is that we focused on sex dimorphism in inflammaging, and the second thing is hormonal cycling, which I'm not going to discuss today due to time constraints. So first, sex dimorphism in inflammaging. So for this particular study, what we did is like we received mice of two different age groups from National Institute of Aging, NIH, and and we have the two different sex, males and females. So what I did is that I extracted the bone marrow and differentiate them into bone marrow derived macrophages with MCSF in the flasks, and then we performed some molecular analysis and also some functional analysis, RNA-seq and phagocytosis, for example. So when we did the RNA-seq, as you can see here, blue being the males and pink being the females, this multidimensional scaling analysis clearly show that sex has like a huge role in having the transcript separated. And as you can see here, there's like 86 different molecules that are significant change in males only, and there's like around more than 500 genes that are modulated only in females with age. So the transcription landscape is very different according to the sex when we age. And we checked some of the genes, and when we did the functional enrichment analysis, so these are some gene sets that are involved in functions like endocytosis, SNOR interaction in vesicular transport, and lysosomes. So this sounds something like there could be some problem with the phagocytosis, this kind of thing. So we did a phagocytosis assay where I took the zymosan, fluorescently tagged particle that isolated from yeast, and then what we did, we put the zymosan with the bone marrow derived macrophages and let it sit there for like an hour. And after that, we checked what's the difference between males and females, and also with the aging. As you can see here, there is no difference, but there is a trend, a decreased trend, but it's not significant. Whereas in the females, there is a significant decrease with the phagocytosis. And as you can see here, with the aged male and aged female, there's also significant decrease. So the phagocytosis decreases with age only in females. So as I did my PhD in AHR, automatically I went back to the RNA-seq and checked what's the level of AHR, and I saw that AHR is decreased with age only in females, like similar trend with AHR transcript. So what we see here is a qPCR confirmation of AHR with an independent cohort. So I show you just one cohort, but this has been tested in 3 independent cohorts, and so far we always had this trend that AHR is decreased with age only in females, as well as the endogenous repressor AHRR is also decreased with age only in females. And moving forward, so what Berenice did is that we wanted to investigate further. So I went back to the literature as usually, and there's like bunch of articles. I just show you some of the articles here, but there's like that, and even recent days, there's even more articles coming that AHR is crucial in modulating immune system. For example, this particular immunity paper where they showed that activation of AHR could direct the monocyte differentials towards the dendritic cells, not towards macrophage. So very interesting. And also AHR is involved in NLRP3 mediated inflammasome and stuff. So it's clearly AHR plays a role in immune system. And what Berenice did is that she took the publicly available data set of AHR ChIP-seq analysis of dendritic cells, and just we compared with the 500 plus genes that are regulated sex-specific only in females with age. So we can clearly see there's a nice overlap between these two and these 171 molecules, 171 genes. So when we check the function of these gene sets, so again, it's mind-blowing that we have this phagocytic vesicle membrane, lysosome, early endosome, and mitochondria. So clearly, AHR is doing something with aging in the females. Going further, we checked the string analysis of protein-protein interaction of AHR, and as you can see here, AHR interacts with ESR1, which is estrogen receptor 1, and several other age-related genes. Here, for example, different genes involved in aging. So conclusion of this particular part is that transcript landscape is different among the sexes in terms of like BMDM transcript landmark. And all females, especially the BMDM, have decreased ability to perform phagocytosis. Phagocytability of BMDM is unchanged in males. So the AHR transcript declines with aging only in females. So now when we think about what's the take-home message, so what am I trying to say is that AHR is clearly doing something with aging, and there's an interaction. If you look at the literature, there's an interaction between estrogen receptor and AHR as well. And putting all the data together, what I'm thinking is that AHR is really, really important in modulating immune system that could potentially be involved in sex-based aging. And also in terms of like this tumor and also in myelination, and putting all together. So AHR, as my title says, that AHR as multifaceted. So basically it has several roles, and it's one of the major proteins that can orchestrate different physiology of cells. And I think that's the take-home message of this this entire talk. And with that, I thank my PhD supervisor, Professor Masad, and his team, and also all our collaborators back in Paris to have this myelination and this tumor study. And then Professor Benayoun and our team back in USC, and all our collaborators for this particular work. And with that, I thank you all for your wonderful, for giving this wonderful opportunity. I particularly thank, again, Professor Subramani, Parni Subramani, and also Professor Chamundeshwari, and all the organizers in this wonderful, wonderful, amazing webinar that you have organized in this particular COVID-19 crisis. And also St. Joseph's College of Engineering for this wonderful platform. Thank you very much.
So thank you, sir. Thank you for an informative presentation on aryl hydrocarbon receptors and how they function, as well as telling us about how phagocytosis is decreases in females compared to males. We have a few questions here, sir. So can we go ahead and answer them? Can you answer them?
So, yeah, please. Yeah, sure.
Okay. So the first question we have here is aryl hydrocarbon a drug target for cancer therapy?
So there's this company called GSK. They are trying to. I think. When I was doing I didn't follow up, but at that time there was like clinical trial level 1, phase 1 with GSK for skin cancer targeting AHR. So AHR, given its multi-function, my personal opinion is that it could not be a great target, but still people are trying, and my patent application is also in that aspect. So it could be a nice target, and we have to be very careful with with a very good candidate. When I say with a very good molecule to modulate the AHR activity, and that is one from GSK. I don't know if there exists some other compounds as well. Some other trial going on.
Okay, sir. And thank you, sir. And the next question is, what are some of the ligands that bind to AHR?
Okay, fantastic question. So dioxin, and there are other they call it as PAS pH, like polyhydrocarbons as well. Bisphenol A, I believe, and these are like some of the xenobiotics, like some toxic elements that bind to AHR, and there's like a bunch of endogenous receptors in our system itself. For example, L-kynurenine. FICC is a tryptophan metabolite. So basically, FICC is formed by UV radiation. When tryptophan is radiated with UV, FICC is formed, which is also an agonist for AHR. There's also IC3, like indole 3-carbinol, which is from this food source. For example, broccolis, this kind of green vegetables, including cruciferous vegetables, has a lot of IC3. And there's like a bunch of paper coming out recent days that the microbiota can have this kind of tryptophan-based metabolites to have increased AHR agonists. And curcumin is also debated to be one of the agonists of AHR. So curcumin, you know that we have that in the turmeric. These are some of the ligands that come to my mind at this point.
So actually there was another question that said, can you tell us some of the dietary sources for the ligands that bind to AHR?
Yeah, that's what I just mentioned. Yeah. So dietary things like indole 3-carbinol comes to my mind at this point, and that's what I said, like broccoli, green vegetables, cruciferous vegetable in general have a lot of IC3. And there is a paper, I remember, that they have supplemented with some broccoli extract or something like that. They are particularly given I3C, indole 3-carbinol, and showed that it could be involved in monocyte differentiation towards dendritic cells. I think it's the immunity paper by Shinde et al. From Canada. Other than this green vegetables and things, I don't know. But curcuma, so the turmeric that we use daily in our cooking as a potential AHR agonist.
Thank you, sir. The next question is, does persistent activation of aryl hydrocarbon receptor signaling result in carcinogenic effects?
Fantastic question, but to be honest,
And can AHR be targeted for other forms of cancer? What will be the efficiency?
Breast cancer. Sorry, I'm sorry to answer. I have no idea. I'm sorry. I don't know. And also in terms of other forms of cancer, I mentioned about this skin cancer. And in terms of this nerve sheath tumor that we didn't translate it further to animal models, and so I left the lab. So, and to be honest, my domain of research in terms of AHR moved a bit away from cancer side, and I'm not a specialist in cancer biology. So for this purpose, we collaborated with Dr. Pasma, who is a clinician, himself an oncologist, and in Curie Institute. So I don't have any idea in this particular question. Sorry.
Oh, so that's okay, sir. The next question is, if a different ligand binds to AHR, is there a possibility of an opposite and a detrimental side effect?
So the thing is, like, the ligand binding is based on the affinity. For example, dioxin found to be the most potent agonist so far. And dioxin is coming from, it's an exogenous. It's coming from, we know that it's an environmental pollutant. And VICGRTB, which is an endogenous ligand, also binds very potently, but it can't compete with dioxin. So let's imagine we have two or a few other ligands given to the same type of cell. I think the binding of ligand to the receptor is primarily based on the affinity and also the dosage. And normally, that could lead to. So the answer to this particular question is that if we have different ligands, do we have some detrimental effects? So to be honest, I tell you, just dioxin, based on the dose, may have different outcomes in different types of tissues. And also for example, for hepatocytes or cardiomyocytes or these kind of things, the TCD dose can be very different. For example, 7-ketocholesterol, which is also an endogenous ligand for AHR, and 7-ketocholesterol. So for example, Schwann cells and these myelinating cells, they're already saturated with a lot of cholesterol in them, so may not respond to 7-ketocholesterol. Whereas if you have VICGRTB, maybe it'll respond further, because I think it's also the biological availability. So in that term, first of all, we need to know how the binding is happening in order to know the outcome. And in terms of like detrimental result, we don't know what's going to happen. And given it behaves very differently in different cell types.
Yes, sir. Thank you, sir. And the last question is, the locomotor deficit caused by the AHR ablation been addressed? If yes, how it could be?
Okay, so basically the question, like if I understood well, so the person who is asking is that if you want to fix this, right? That's the thing, right?
Yes, I think so, sir.
Okay. So, I mean, I didn't show all the results, but if I understood the question right, I mean, please correct me if I'm wrong. What I understood is that, so basically you ablate the AHR, we have the locomotor deficits. And if you want to address this problem, how could you address this problem? So basically this loss of function or gain of function, this kind of knockout studies are there to understand what's the physiological role of a particular gene, and in order to explore if it's cell autonomous or non-autonomous, we use tissue-specific or cell-specific knockouts. So basically, in normal physiology, we have AHR, and this myelin development is occurring in a normal way. So there wouldn't be any locomotor problem. And this is the reason why we went to see the development. If you don't have AHR, the development is happening normally or not. At the same time, we also checked the pathological case. That's the reason we checked, okay, maybe the activation of AHR could have some detrimental effects, and that was the case. If we have to address, I would just do rescue experiment not overexpressing, basically having the same dose of AHR protein somehow with gene therapy in order to rescue the locomotor deficit. But we didn't do the experiment, so we can't know what could be the outcome. But that could be one of the strategies that we can try.
Okay, so thank you, sir. Actually, I have a question. It's not a technical one, but I saw a slide in your presentation and it had a logo that said Mayava. Can you explain further about it?
Oh, fantastic. So thank you very much for asking this, Swatika. So a friend of mine from now is a postdoc in the University of McGill, Ayapasamy. So Dr. Ayapasamy and I, we always wanted to have some joint venture, particularly starting a YouTube channel long back when we were in France. And he also did his PhD in France, but in a different city, Montpellier. So, and during this lockdown, we wanted to materialize. It's not we wanted to materialize. I think we thought it's the right time to materialize, and we started a YouTube channel, ironically, on a Fool's Day, 1st of April. And it's performing really well. And the name of the YouTube channel is Mayava. And Mayava is basically, we wanted to create awareness among the Tamil community that if you hear some news or some scientific data or something, please just don't take it for granted. You have to have some skeptical thinking and put your knowledge and do more research and do your own research and come up with a conclusion. Don't say this particular YouTube channel, which is one of the top most YouTube channel or the top YouTube channel in Tamil, if he says that could be correct. No, not really. So put all the information that you hear into analysis and analyze them with your own knowledge and further knowledge and make some decisions. So this is where we are now. And yeah, thank you very much, Swatika, for asking this. And this is the reason why we started Mayava. It's a YouTube channel. Participants or someone who is watching this YouTube live or later on, you can please go check out our channel. Just type in MEIYAVA and Mayava, and you can find us. And please check out our contents, and then you are welcome to comment if we did something correct or something wrong, and we are open to accept those comments as well. And please do subscribe to our channel.
Oh yes, sir, definitely, sir. So I'd like to thank you once again for taking the time to join us today and imparting us with your knowledge on AHR, sex dimorphism, and phagocytosis. Also, we appreciate you taking the effort to debunk various incorrect news via your YouTube channel. We will definitely check it out, and I urge the other participants to do it so as well. I'd also like to take this time to thank our patrons, our HOD, Dr. G. Srikumar, our convener, Dr. M. Chamundeshwari ma'am, and our co-convener, Ms. K. R. Preeti, for giving us the support and making this event possible. And finally, I thank all the participants for your cooperation and ensuring that the event was seamless. Our last session to conclude this virtual international conference is on engineering semi-synthetic regulons in yeast for non-native nutrient assimilation and will be given by Dr. Venkatesh Endalur Gopinayaran, who is a scientist at Massachusetts, USA. That session will be this evening at 4 p.m. We appreciate you all for being here. Stay safe, and I hope to see you all in our last session this evening. Thank you, everyone. And thank you, sir.
Thank you very much, Swatika, for this wonderful introduction. And thank you all for participating and all the faculty members for this opportunity. Thank you very much.